My name is Janos Filep. I'm professor of pathology and cell biology from the University of Montreal. It's Montreal in Canada. That I have just made a presentation to the International Academy of Cardiology Annual Scientific Session 2015. My topic was on C-Reactive protein, and that is how this protein may regulate the inflammatory process underlying acute coronary artery disease. C-Reactive protein has been implicated as a predictor of cardiovascular event, though that this issue has been very much of a controversial. Even recent studies that indicate that CRP may guide therapy as suggested by the Jupiter trial. However, the function of this molecule is still remain largely unknown and controversial. We propose sequential changes that this molecule undergoing conformational changes which expression of different biological activities. The conformational changes that start with that C-reactive protein, that this is a pentameric protein present in the plasma, which predominantly have anti-inflammatory and protective roles. So that is part of the innate defense mechanism. However, when this molecule undergoing dissociation structurally became modified or dissociated into its subunits, for example, by the surface of activated platelets or apoptotic cells, that this results in expression of another type of pro-inflammatory activities, including activation of endothelial cells, leukocytes, and activation of platelets. However, that this changes renders the molecule susceptible to cleavage by proteases released from activated leukocytes, for example, by neutrophils, yielding to the release of bioactive protein. One of these bioactive peptides. One of these peptides, this is a C-terminal hexapeptide called peptide 201-206, which we found that is exert potent anti-inflammatory and antithrombotic activity. So that we think that this is like these sequential changes in CRP structure that would lead expression of a varying and often opposing biological action, which may fine-tune the inflammatory process underlying cardiovascular diseases. On the other hand, that this data is the intriguing possibility of targeting formation of structurally modified mCRP as a potential target to diminish the inflammatory process, or using C-reactive protein-derived peptide as potential therapeutics to combat the inflammation underlying acute coronary artery syndromes.